All right, welcome back. So this video is going to be for the Thursday, so day 2 of our first week of labs. So if we look at the syllabus, it says results from previous labs and streak plating. Well, I'm recording all of these videos, well, most of these videos on a single day, so I'm not going to have bacterial growth to show you in these videos, but what I will do is I'll post pictures of them once I do have the results of those available. So in lab, you'll be looking at your results and then doing streak plates, which is 1-4. So streak plating is another really important technique that microbiology students need to learn, as well as if you work in a microbiology lab. It is very, very common. So you learned aseptic transfer on Tuesday, you'll be learning streak plating today. Okay, so streak plating, this is gonna be lab 1-4. This is the pattern that you'll eventually want to learn how to do. So what you can observe is we see a whole lot of thick bacterial growth along the edges of the plate, and eventually towards this middle open spot, we can see individual bacteria. Okay, so you can use your lab manual to see that picture. So the goal of streak plating is to isolate colonies. So an individual colony is started by what's called a colony forming unit, or CFU, which is basically one cell or a few cells stuck together. But what it means is all of the bacteria that are growing in that one colony came from one original source. So technically, barring any mutations, they are all genetically unique. That's important in microbiology because if you're going to be doing experiments, you don't want to take bacteria from a mix of where genes could be different. You want to take your sample from a specimen that is genetically identical to each other. So each student in this lab will be making 2 streak plates. You'll get an opportunity to practice this technique twice today, but we will be using it quite a bit more throughout the rest of the term. Okay. So in the diagram, there's some pictorial images to help you with that and your instructions, and also a drawing or a diagram kind of showing you the steps. We're going to focus on a 4-step streak, okay, in class. We are not going to be doing the T streak, so you don't have to worry about that. And then there is kind of a blank petri dish on the back here that you can use a light pencil if you want to practice your pattern before you actually get the bacteria onto the petri dish. And I'm going to just be giving you some tips and tricks on how to best do a streak plate. So again, I'm going to get bacteria out, so I'm going to sterilize my table, get some bleach, make sure the workspace that I'm using is nice and clean. I don't want contaminants. Contaminants show up a lot easier on those petri dishes because the lid tends to stay open a lot more than those tubes, and we cannot flame the petri dishes. So I'm going to start off with my 1 petri dish. I'm going to label it. I still got my 1 M. flei critter that we have here. So I'm going to label M. flei. Grows at 37 degrees. Today's still the 9th. This is TSA, and I am CLA. So I've got all my 5 bits of information on the agar side, so I know that that information is going to stay with my bacteria. Okay, I'm going to get my burner set up. So I've got my burner in my gas jet. I got my striker ready to go. There we go. Turn down our jet engine. So now I'm ready to go. I have my bacteria in my test tube rack. I've got my labeled plate, and I've got my loop. Okay, all the tools that I need, I'm really organized. So what we do first, we're going to flame my loop, make sure I have it nice and sterile. The first streak on a streak plate is going to be your thickest bacteria. Okay, so as I let this cool down, again, we're going to aseptically, the best that we can, transfer bacteria out of my culture tube, where my bacteria is, onto the plate. My first streak is going to be right up next to the edge of the petri dish. So here I'm going to flame the neck of my tube. I'm going to grab some bacteria, flame the neck of my tube, cap that, put that back into the rack. All right, so I'm done there. So I've got my bacteria, right, my little bacteria there. So then I'm going to pick up my plate. I like to do it this way, keep the lid on the table, pick up the plate and find an edge and just go back and forth very gently. Just kind of squiggle back and forth. And if you'll notice, my loop is not, I'm going to finish this real quick and then I'll show you what your loop should not be. Okay, so I've got my nice little streak. I don't know if you can see it. All right, there you can kind of see some of the lines. Streak, streak, streak. This is our first streak, way up to the edges. Put that back down. So let me flame my loop. So there's a couple of do not do's here when we're doing streak plating. So whenever you have a Petri dish, you don't just want to leave it open while you're doing other things, because from Ubiquity of Microbes, we'll be learning that stuff can fling into and contaminate your plate. So you could either lift up the lid, do your streak, and put down the lid, or how I prefer to do it, but this is my preference, is I like to leave the lid on the table, pick up the plate, do my streak, and then put the plate back down, auger side up like this. So if anything catches on the lid, it's gonna stay on the lid. It's never gonna land on the surface of the auger, because remember, these are always incubated this way too. So anything that lands on the lid is just gonna stay in the lid, as long as you don't turn it upside down, okay? The other no-no that I wanna point out is how you, when you push down on your loop, I'll use my hand as an example, move the burner out of the way so I don't burn myself, is we want the loop to just be kind of like this. We should never see the loop bent like this. That's too much pressure. You can cut into your auger and it bends your loop, right? So we want to keep our loop nice and straight. Whenever we're touching, it's a really flat angle, and we're just going to go back and forth like this. And if you'll notice, my loop never is even bending. So you really shouldn't ever put pressure on it enough to you're going to bend your loop. And they mention that here in the instructions. Okay? You don't want to be too vertical, because that can cut the auger. It's really delicate. We don't want to be too hard, because we're going to bend the loop and that could cut the auger. We want to be Goldilocks just right in the middle. So then the next streak that I do, I want you to watch my loop, and so you can see how gentle I am actually putting pressure on the loop as I'm streaking. So we're going to flame the loop again. You will have already had it flamed because you didn't touch it to your hand and glove and stuff. So when we're streak plating, we always flame in between every streak. We grab other bacteria the first time and we never go back into the tube. We're done with the bacteria. All the bacteria we need is in that first streak, what I put on the top. So once I see that, I'm gonna turn it about a quarter turn. So this is where it was. I'm gonna do a quarter turn. And so now my bacteria is right here. So I'm gonna take my loop and I'm gonna streak a few times, dragging some of this bacteria into this new space over here, okay? So watch my loop. So I'm gonna go 1, 2, 3, 4, and then I'm going to squiggle again, but I'm not going back into that first streak. I've only went back there 2 to 5 times. You know, you just don't wanna drag too much of it over. And then now I've spread that new bacteria into this next section, this next quadrant. Put my plate down, flame my loop, okay? Let that cool down. I'm gonna do another quarter turn. And now I'm going to, in that second streak, I'm gonna go a couple times here. So I'm gonna find where I left off. I'm gonna go 1, 2, 3, and then I'm gonna drag that over into that next blank area. I know you probably cannot see this in the video. It's hard to see even in real life, just because bacteria is invisible, but you can see your streak lines. Okay, so I'm gonna do my last quarter turn. This is my 4th streak. So I've done 1, 2, 3, kind of boxing out. This big middle spot is completely open. Take my blue glove. So I have my first streak, my second streak, my third streak. So now for my 4th streak, I'm gonna again go 1, 2, 3, and then I'm gonna spread them out nice and big zigzag right into the middle there. Okay, put that down, flame my loop, and then you're done. So that is your 1 streak plate. So they don't take very long. It's a little bit challenging because you cannot see the bacteria. It's not like you're drawing a line on a page. So you have to use kind of the reflection of the lights on the surface of the auger to see where your streak lines are. And then just make sure you're following those diagrams and the guidance that's in the lab manual right here. So if you'll notice kind of our first 1, 2, 3, and then the 4th takes up that nice big space in the middle. And our goal is to isolate colonies to see if we get individual isolated CFUs. All right, so that is the end of our streak plating. I will see you next time. Bye.